Hi everyone. I'm Magdalene. I'm the coordinator for Upper Snowy Landcare Network, and also recently the project manager alongside Sonya Duus on Landcare's Climate Ready Revegetation project under our Phase 3 project. First up, we've got Matthew Brookhouse from the ANU Fenner School of Environment and Society. Following on from Matthew, we'll have Tricia Hogbin from the Research Centre for Ecosystem Resilience. And then the last presenter will be Sonya Duus with the Yass Area Landcare Network, who'll be presenting on their Climate Ready project. So I will jump straight to Matthew.
Thank you very much. I'm going to share a set of slides with everyone. And while I turn that on, give me a moment, you should be able to see, oh, that's my last slide. Let's go back to the beginning. While that's happening, I'm very happy to be here on Ngunnawal country tonight. I imagine like in a lot of places locally, it's a cold and it's finally feeling like winter. And that's particularly important when it comes to an area of work that I'm involved in pretty routinely now, and that is work on high elevation snow gum. Now, high elevation snow gum is not necessarily something that falls generally within perhaps a land care domain, but it is a domain in which we are very much focused on because of a phenomenon that's playing out insofar as an outbreak of insects that are impacting across all of the Australian Alps at this point in time. And it is presenting to us questions, and we are responding to and taking on questions around how we respond at an entire landscape level. And one of those responses is thinking about how we respond in a climate ready sort of framework, and in particular, how we select for taxa that are appropriate replacement for what we're seeing at high elevation. So to focus on the problem, let's first think about how what climate ready really means, and the way that we might normally think of being climate ready or climate taking on a climate adjusted provenancing approach. Now, first of all, we need to acknowledge that climate is shifting, and there have been substantial changes in temperature in the recent decades. And in response to the speed of that change, we need to think about the plant materials and the way that we respond inside reserve areas and also outside those. Insofar as restoring affected ecosystems impacted by fire or by what we see as an unforeseen selective agent, we need to think about alternative taxa or alternative genetic material that may not be the resident material that is in place. Now, there are risks that come with that, of course, and they relate to the way that we might select. But one thing to take on board at this stage is while we often think about the impact of climate as being something that is purely abiotic, there are these unforeseen selective agents insofar as insects that have capacity to respond. So biotic agents have a far greater capacity to respond to change. And that's the kind of change that we are likely to see and are seeing happening at a far faster rate than trees or vegetation can possibly respond. So the question then becomes, how do we respond from a climate ready perspective to these unforeseen selective agents? Well, let's understand the problem a little bit more and focus on what snowgum dieback is, because that's the problem that we're confronting at a very large scale now. Now, this image on the left-hand side is really the type image of wood borer infestation in snowgum. That image is just near Guthiga, but as I've already indicated, this is a problem we're not just seeing in Kosciuszko. This is pretty widespread. It's here in the ACT, it's in Victoria, and in the top right-hand corner, you can see that's Eucalyptus lacrymans, a very narrowly distributed species that is entirely affected across its entire distribution. So it's a substantial problem, and it is one that is related to or is the product of an insect, but an insect that's very responsive to changes in the host material as well as in climate directly. Now, this is an indication of if we focus on just one of the snow gum taxa, we know that snow gum is made up of multiple subspecies. Here, Eucalyptus pauciflora nephophylla. If we look at its distribution taken from, or records of its distribution taken from the Atlas of Living Australia, some of those are obviously planted specimens like those down in the Latrobe Valley. But if we take a map of those observations and then overlay on top of that the observations of dieback caused by beetles across that distribution, we can see that all of the range of Eucalyptus nephophylla is impacted. Now, what does that actually look like out in the field? Now, from a distance, this is the type of thing that we would see. So again, this is just near Guthiga, and something's very striking here, and that is evident in the graph as well as it is in this little bit of footage. And that is that as you climb in elevation within Eucalyptus pauciflora nephophylla woodlands, the severity of the impacts decline. Now, I was very careful in sort of phrasing that, and that is because it is within Eucalyptus pauciflora nephophylla, not in the lower elevation pauciflora. So there's a subspecies boundary at about 1600 meters, and it is at the lower elevation of the higher elevation subspecies where the problems really kick in. Now, that suggests to us immediately that there may well be a genetic underpinning that is responsible for that difference between pauciflora and nephophylla subspecies insofar as the impact of dieback. And we've spent quite a bit of time in the last 5-6 years working through that. And this is just one example of that. You can see these three panels taken from a nice piece of work by Cal Bryant here at the ANU, in which Cal looked at a whole suite of traits. And here I've just picked out three key ones: leaf size, the Huber value that represents the amount of sapwood to leaf area, and then the leaf mass per unit area. Now, the key message here is that as you move up in elevation, and we've kind of lost the x-axis there that goes from about 1,000 meters in elevation up to 1,800. As we move along elevation, we see a very strong change in leaf traits. And I'll just sort of highlight that if you're sort of missing it. Just at that point, there's a clear break point. And that break point is where we change from pauciflora into nephophylla in terms of subspecies. There's a very strong change. Now, the only way you can really achieve these kind of changes at a tree level, especially that change in the middle where we have more sapwood area, more conductive area in the stem to the amount of conductive area in the canopy, is that you increase the amount of water that the plant is using. And of course, that is a strategy that high elevation snow gum has, not so that it can use more water, but rather so that it can take in more carbon and then accumulate more sugars to get it through a shorter growing season. So it's a key adaptation that underpins the very reason why nephophylla is there, and it underpins the plant vigor itself. So if we took a strategy in which we selected for plant vigor at high elevation, in this case, we would select for a trait that benefits these characters here. This is Phoracantha master's eye. This is the species that impacts upon snow gum, and it benefits from, as you can see on the right-hand side, as the bark desaturates a little bit, the survival of the insects really shoots up. So you don't need to be a drought-stressed tree to achieve this outcome. You just need to be a tree that desaturates a little bit during the day, and that happens more or less every day during summer. So why hasn't it happened in the past? If that's happened all the time, well, the answer is pretty straightforward, and that's why we are so concerned right now. This is a reconstruction based upon our temperature modelling that we have built over the last 5-6 years of when adult Phoracantha beetles emerge from their pupation chambers, set out to mate, oviposit, and then the new neonates get stuck into the next round of trees. What you see in the top here is how the seasonal accumulated heat has risen over the last 2 decades quite noticeably, and associated with that, the emergence of insects has crept progressively forward. And we can see from the data that we have in front of us and our modelling that every increase in mean annual temperature of 1 degree brings the emergence of adults forward anything up to 18 days and then really opens up a window for the insects to affect trees. So we have a problem. The problem is that we have a heat-driven expansion of the range of Phoracanth. We have a host that is completely naive to the pressure that Phoracanth bring because in the past Phoracanth were not present at the levels that they are now. And because they were not present, they don't have a defense. They have developed a life history strategy to profit and do well at high elevation. And that is the very strategy that is underpinning the problem for them. So in short, snow gum is not stressed. And so looking for a trait that reflects plant stress is not going to help in this case. Rather, it is being impacted by a heat-sensitive agent that is being benefited by increasing temperature in much the same way that snow gum is. And you can see in snow gum on that bottom panel, growth increases substantially as the seasons warm. And we can see in the middle of that black and white panel, in reverse, we can see a sequence of very warm years that produce not only wide tree rings, but tree rings that have a lot of late wood that have a lot of embedded energy. So where are we in terms of responding? Well, there are a lot of things going on, but one of the big ones, and I was only discussing these trials today, one of the big ones is to both understand not just the traits that underpin dieback, but then also thinking about alternative sources of genetic material that are out in the landscape to bring into trial plantings. And there's a whole body of work here supported by a number of ARC linkage grants, but a really key component of that is slightly hazed out at this point in time. And I'll skip to the next slide because that's the important bit, is that all of this work is taking on a collaborative approach together with the parks agencies themselves. There's no way of putting this nicely, and that is we are dealing with a high elevation forest community that a lot of people place a great deal of value on. And we are, during meetings and in management discussions, talking about the progressive transition away from the existing taxa that are present at high elevation. And that's a confronting issue. It's not something that's going to happen without a lot of management support. And so selecting and working towards a replacement or a climate ready planting or an adaptation process is one thing, but having everyone on board is another. I'm going to stop there. I could talk about snow gum dieback all night and then you would still hear me talking about it tomorrow morning. But I'm going to stop there and leave time for Tricia and the other speakers, Sonya, to speak. But I'll be here for the rest of the meeting as well.
Okay, so I'm joining you today from Wonnarua country, and I'm going to be talking about how you can use the Restore and Renew web tool to guide seed sourcing for resilient climate-ready restoration. So I'm with the Research Centre for Ecosystem Resilience at the Botanic Gardens of Sydney, and we're a team of 20 multidisciplinary researchers who all have a common interest in guiding restoration and conservation of resilient ecosystems, hence the name, through large-scale genomic projects. And to give you an idea of the scale of these projects, so us and our collaborators have collected more than 109,000 leaf samples from more than 2,000 species across Eastern Australia. And so far, 62,000 of those have been sequenced, and they all contribute to one whopping big data set we call our genomic knowledge infrastructure, which can answer all sorts of wonderful questions now and into the future. Now, one of those applications is what we're talking about today, the Restore and Renew web tool, which is freely providing genetically informed seed sourcing guidelines for 100 species so far. And the capacity is there for that to hopefully grow to thousands of species across Australia. So what I'm going to be talking about today is the start of an ongoing collaboration that's all about helping Southeast Landcare restore resilient climate-ready ecosystems. So what I'm going to be talking about this evening is we're going to be reminding ourselves what is a climate ready ecosystem and why the genetic diversity of single species is relevant to that. I'm going to be talking about why it's time to move on from generalizations regarding local provenance to evidence-based genetic neighborhoods, and that it's time to shift our focus from plants in the ground to restoring plant populations. I'm going to introduce you to the Restore and Renew web tool and how it can guide sourcing of climate-ready, genetically diverse material for restoration. And then we'll finish with a web tool demonstration. And as Magdalene mentioned, there'll be an opportunity for you to dive deeper in subsequent workshops and mentoring in how to apply the guidance to your needs, be they regional planning, seed banking, seed production areas, and so on. So what I'm hoping today is you go away understanding that the plants you put in the ground are going to affect not only the short-term survival of your restoration and whether or not those plants survive, but also the long-term survival or success of your restoration. In other words, whether or not these plants produce self-sustaining populations that become part of a resilient ecosystem. So what is a resilient ecosystem? So an ecosystem, a community of living biotic things interacting with each other in their non-living abiotic environment, and it's made up of populations of different species. So for example, we have a grassy woodland near Goulburn, Marlene, one of my colleagues, and we have this acacia in the foreground, Acacia paradoxa. It's just one species. We have many individuals of the same species growing in the same location at the same time, interbreeding freely and producing offspring that replace those that senesce, a population. We also have a euc that's interbreeding and producing offspring that need to be able to adapt to changing environments. So their offspring need to be able to adapt to changing environments if this ecosystem is going to persist. So to restore an ecosystem, the populations of species within need to be able to survive, reproduce, and maintain themselves. In other words, they need to be self-sustaining. So let's look at that idea of self-sustaining in a little bit more detail. So for a restoration to be self-sustaining, it needs to be made up of functioning and resilient populations of its component species, and they need to be fecund, reproducing now and into the future, adapted to current conditions, and adaptable to future conditions. So unfortunately, a lot of the emphasis in restoration to now has been on the quantity of the plants going into ground and not necessarily the quality, which is really important because if we want to see plant populations that can reproduce and adapt to change, they need genetic diversity. Because genetic diversity, and we'll talk about this in more detail now, genetic diversity maximizes adaptive potential and minimizes inbreeding. So looking at adaptive capacity, the more genetic diversity a population has, the more likely that at least some individual within that population has a trait that enables it to adapt to change, or to use Matt's term, the unforeseen selective agents. So we know populations with really narrow or low genetic diversity are less likely to have individuals with a genetic makeup that enable them to adapt, and they're highly vulnerable to future threats, such as here we have Phytophthora, curl leaf disease, myrtle rust, climate change, and so on. Whereas if you have, and even think of the, you studied in high school biology, the potato famine. So we know that populations with more genetic diversity are more likely to have some individuals that are able to adapt to change, and therefore they're less vulnerable. Now let's look at the second issue that's important, inbreeding, and why it's not something you want to see in your restored populations. So inbreeding is with individuals that are genetically closely related or genetically similar mate and produce offspring. So it's a seed from the same tree or from the same population or from a small population. So unfortunately, current practices in restoration encourage inbreeding. For example, in eucs, it's super easy to collect thousands of seed from a single branch from a single tree. And these are all siblings. They're all closely related. So we don't want to see them. And that's unfortunately what we do see is we see hundreds, if not thousands of individuals all sourced from one plant growing close to each other. So let's give ourselves a bit of a genetics 101 refresher to understand the potential consequences of this planting siblings next to each other. So alleles, alternative versions of a gene at a given location in the genetic code. Each individual has two, one from each parent. They can be homozygous or heterozygous, and the dominant allele is expressed no matter what. But the recessive allele is only expressed if it was in the homozygous state and not masked by a dominant allele. So unfortunately, mating among close relatives increases homozygosity, decreases heterozygosity, and increases that expression of deleterious recessive alleles. Those alleles that usually exist in a population and aren't selected against because they're hidden. So we lose that heterozygous advantage, and it can result in inbreeding depression. So that's where populations may not produce seed, or the seedlings may be weak and unlikely to thrive. And that's not something we want in our restored populations. So to restore resilient, climate-ready ecosystems, we want to strive to restore self-sustaining populations for each of your target species. And keep in mind that may mean that you're actually focusing on or targeting fewer species, but better quality material, because it's really important. We want to capture that diversity within species to facilitate resilience or that adaptive capacity. But we also want to remember that we want to strive for diversity within individuals. In other words, we don't want all our individuals from a single site or from to be siblings and closely related, because we want to avoid inbreeding depression. But unfortunately, most of them, or much of the material used in restoration these days typically has low genetic diversity due to two main reasons. Habitat loss, so we've already lost a lot of vegetation. We know as population size decreases, we lose genetic diversity. And also, it's amplified by narrow seed collection. So to give a schematic representation for a single species, we have a genetically diverse natural population. Come along, we lose half of it, and just by chance we lose genetic diversity. And then we can actually amplify that loss of genetic diversity by coming along and collecting seed from only one or few individuals. So in the short term, it may look like it was a successful project because those plants are surviving, but they're actually not producing new recruits because of high levels of inbreeding. Now, we could do slightly better. You could come along and collect material from a slightly broader number of individuals. And in the short term, you think, I've done really well. Look at these new recruits coming along. But then a new stress comes along, for example, climate change, disease, or an insect. And because of the low genetic diversity, just by chance alone, they're less likely to have some individuals that can adapt to that threat. So they have low adaptive potential, and the project fails in the long term. So what we want is, even in fragmented landscapes where we've increased isolation and we've lost genetic diversity within population, we want to collect a genetically diverse seed collection so that we have genetically diverse restored populations, so that we see that normal recruitment in the short term. But also we want in the long term for our restored populations to be self-sustaining and adaptable. So this is probably a really good opportunity for us to pause, and I want you to revisit the whole idea of what is local provenance and why have we been told to use it. So I want you to share the 1 or 2 sentence answer briefly in the chat. What is local provenance? We've been told to focus on local provenance for decades, and why have we been told to use it? I'll wait until I can see 1 or 2 answers. There you go. Wonderful. So while you're typing those in, I'm actually going to keep moving on. I've just lost the ability. I closed my screen to look at your chat, and I've got all confused, and I can't move my slides momentarily, so this might just take me a minute to fix this. I'm not quite sure what's going on, but I'm going to stop looking at the chat and try and see if I can get this working again. Eek. No. Okay, I'm going to close something. It shows a clapped. I don't know why I cannot see. I must have pressed something whilst I was looking at your chat, and I'm going to stop sharing my screen and come back again and start again. Share. Okay. And it's still not moving. Stop share. Okay. Now I'll share. Okay, so this is going to work. Okay, so sorry about that. I gave you time to answer questions. So really, and I could see some of the questions coming through, and really, local provenance is all about, it's a generalization aimed at capturing material adapted to local conditions. But we all know local conditions are not constant. They're changing due to climate change, threats, habitat disturbance, and so on. And also, it's aimed at avoiding unintentional consequences of mixing individuals that are too dissimilar. So I want to very briefly touch on what this may mean, is that we don't know what the consequences, Matthew spoke about, looking at, spoke about different subspecies. One is able to tolerate the insect, another isn't. We could look at mixing those subspecies. We don't actually know what the consequences of mixing these genetically dissimilar individuals is, though there are some common consequences, one of which is the breakup of co-adapted gene complexes. So these are groups of genes, if you think of a DNA sequence, and they've evolved to be confer some sort of fitness advantage on the population. And in a population where gene flow is being shared, those co-adapted gene complexes are maintained. But then if you bring in an individual that's genetically dissimilar, you can actually break apart those co-adapted gene complexes, and then you lose that local adaptation. That's one consequence. But we don't know how far is too far. Like, how far is too far in terms of mixing these unrelated individuals? So, if I ask in a presentation, what is local provenance? Some people may say 4 kilometres or 40 kilometres or a local government area or a catchment. We know that we actually can't generalise about how far is too far. So here's an example, the paper there, QR code. It's for 5, and I'm not trying to get my little cursor back because I don't want to mess up the mouse, but we have 5 co-occurring acacias. They all have a similar geographic distribution, and this paper shows the grey area is a genetic neighbourhood. It's effectively a local provenance, but it's based on genetic data rather than assumptions. And so for Acacia swabiolens and Acacia ulicifolia, they have relatively small genetic neighbourhoods, whereas Acacia linifolia, longifolia, and terminalis, their genetic neighbourhoods are much larger. So a genetic collection area is similar to local provenance, but it's based on genetic data rather than generalizations. So it's also called genetic neighborhoods, seed transfer zones, and so on. So they're an area across which you can safely source material to maximize genetic diversity and avoid unintentional consequences of crossing individuals that are too similar, because they're already sharing genetic material across that area, either in the past prior to habitat, either currently or prior to habitat loss. So this is the information, that genetic neighbourhood or genetic collection area, that Restore and Renew gives you for 100 species so far. There's a link to the web tool, but a really easy way to find it is to go to the Adapt New South Wales website, click on the Resources tab, and under the tab, among the NARCM guidance and so on, is the Restore and Renew web tool. So in the subsequent slides, I'm going to click on a map and give you an example of a reserve in the Illawarra, east of Albion Park Rail, and I can't see my notes anywhere, so I don't remember the name of the reserve, but some of you are familiar with it. And so Illawarra subtropical rainforest is actually, we only have about 15% left of this particular plant community type. So we know if we're going to be collecting seed locally, only from the Illawarra, for example, from those isolated remnants, we've already lost genetic diversity due to random genetic drift, bottlenecks, and inbreeding, and so on. So Restore and Renew can help you restore self-sustaining populations in environments that are heavily fragmented, where we're dealing with not being able to get genetically diverse material. So what the web tool does is it synthesizes the latest genetic and climate science data to help you design evidence-based resilient restoration. So behind the really user-friendly interface is the DNA data for more than 60,000 plants and also the NARCLIM 2.0 modeling. And for each restoration site, so this dot on the map is for the restoration site near Albion Park Rail. So for each restoration site, it identifies species-specific genetic collection areas. So these are areas across which gene flow naturally occurs for a particular species or did prior to habitat loss and fragmentation. So in this example, it's the Illawarra flame tree. The pink dots are its natural distribution. And so this species genetic collection area, or the area across which gene flow occurs naturally, is kind of south of Port Macquarie to the southern extent of the distribution of the species. So you know that if you're collecting material from within that area to maximize genetic diversity, you're actually aligning with a natural process. You're not actually doing anything that wouldn't be happening naturally. So that's the genetic collection area. This is for the Illawarra flame tree. What we can see, here's another example. Again, if you're going to restore that site, Port Jackson fig might be one of the species you might choose. It has a much larger genetic collection area, all the way from Townsville to the southern extent of the distribution of the species. So we know, based on the genetic data, that this species is or was sharing gene flow across this area. So there's going to be no negative consequences of mixing this material because it's already occurring naturally. Or another example, the common Wilkia has a much smaller genetic collection area south, so you wouldn't be incorporating material south of, north of Gosford, sorry. So the web tool also identifies future climate match sites. So these are in orange, and this is identified for that particular restoration site. The areas in orange are currently experiencing climatic conditions similar to what the restoration site is projected to experience in the future. So material sourced from these areas is already climate ready because it's already experiencing that future climate. So if you source seed from there, it can be considered climate ready. So you can use the web tool to design genetically diverse climate ready restoration by considering both the genetic collection area and the future climate. So here's a schematic representation of a hypothetical collection strategy for Illawarra flame tree. For that Illawarra restoration site, we have 5 packets of seed. Ideally, each of these would be 5 individual packets of seed or a handful of seeds from multiple maternal lines, and they've all been maintained. And so you want to maximize genetic diversity by collecting from multiple plants at multiple sites across the entire genetic collection area. And you can further support the climate resilience by making sure at least some of those collections come from those future climate areas. So what are some of the wonderfully worthwhile things the Restore and Renew web tool can help you to do is to recover genetic diversity and reinstate gene flow that's been disrupted by habitat loss and fragmentation. You can bring that genetic diversity back together again. You can move beyond generalizations regarding local provenance and source seed from genetic collection areas that align with natural gene flow. You can enhance climate readiness by sourcing material from areas already experiencing climatic conditions similar to what your restoration site is projected to experience in the future. And you can avoid wasting time and money on failed restoration by shifting the focus from quantity to quality and from plants in the ground to self-sustaining populations. You can minimize inbreeding and enhance reproductive fitness. You can maximize adaptive potential and increase the chance of restored populations being able to adapt. And you can, and this is one we haven't touched on yet, but it's a really important one. You can also overcome the limits to seed availability by discovering that for many species, the area from which seed can be safely stored is much bigger than what you may have anticipated when you're thinking about what is, local provenance. So that means that the pool of seed that is available to you is typically much larger. So species available on the web tool so far, there's 100 species so far. The capacity or infrastructure for many more. We have some species that are very close to being uploaded, and hopefully one day we have funding to be able to put many more species in there, because we have the process or the workflow and the infrastructure, or they're ready to go. So we can put an infinite number of species on the web tool. I just wanted to touch on 2 species that are close to being updated to give you a bit more information or context on what the data is behind the web tool. So for example, this is forest red gum or Eucalyptus teraticornis. The grey dots are herbarium records, indicate the distribution of the species. The purple dots are where we've collected leaf material. Each of these dots, it's 6 individuals, and that's been sent off for sequencing. And the orange dots are sitting in our storage and haven't yet gone off for sequencing. But we can aim to capture a relatively good spread of populations across the entire distribution within the target area. And there's other species like Eucalyptus moricana. We need to do a lot more sampling in the western part of the species range. So I wanted to briefly, before we jumped into the web tool, I wanted to briefly chat about 2 more ways you can further maximize success. An important one is to not to pull provenances and to maintain maternal lines. So seed collections can be better used in restoration if we keep the seed from different sites, and ideally different mother plants separate, when collecting, storing, and propagating. Because as you can imagine, all the seed from one plant or from one population are all closely related. You don't want to be planting those out altogether. What we want to do is we want to keep them separate in the nursery and in the seed storage, etc., so that when we plant them, we can actually mix them to maximize outcrossing and minimize inbreeding. And also importantly, it allows you to isolate hybrid, inbred, or planted maternal lines. So I guess we've looked at enough commercial seed packets to know that hybridization, high levels of kinship or inbreeding, misidentifications, it's all very common. Keeping those maternal lines separate makes it easy to pull them apart. This paper there talks about 10 reasons why it's a good idea. One example from that paper, river red gum, Eucalyptus camelliadensis, we had 10 mother trees in Wallenby Brook. We sequenced the mothers and the seed, and what we found is that there was one mother in purple up here that clustered with material from further out west that we subsequently found out was planted and looked natural. There was only probably a few or number of founder individuals, low genetic diversity. Eucalypts hate to self, so rather than self, they outcross. So those seed were all hybrids, camelliadensis and amplifolia. So by maintaining maternal lines, we were able to pull those hybrid individuals out of the plantings. And we also found another individual where all the seed was highly inbred, multiple generations of selfing. So again, we could, by identifying those, we could use them appropriately. Now, another important point, this might be obvious to most of you, but don't restrict it. So if you want to restore, a particular plant community type, don't restrict yourself to sourcing seed only from that plant community type, because although it's a really easy way to petition floristic diversity, it does not reflect the evolutionary history. Plants don't pay attention to those plant community type boundaries, and by unnecessarily restricting yourself to only the same community as what you're restoring, you're decreasing the available genetic diversity, and there's a paper there that talks about that. So in summary, remember, you're restoring self-sustaining populations, and so you need to sensibly maximize genetic diversity, and you can use the Restore and Renew web tool to do that. You want to grow your plants from seeds collected from multiple plants across multiple sites within the genetic collection area, including sites that match predicted future conditions. And you want to keep seed from different sites and ideally different plants separate during propagation so that you can mix them together at planting to maximize outcrossing, and don't restrict yourself to sourcing seed from only the same plant community type. Just quickly before we jump into the web tool, stay in touch. So my email address was in the first slide. The RISA homepage, you can subscribe to our quarterly newsletter. We produce an annual publications booklet that brings together all our abstracts that are very relevant to conservation and restoration, and you can see some stories there we've written as well. So I'm going to jump into the web tool now, and hopefully when I quickly shared my screen before, so I'll get someone to jump in and tell me that you can see the correct. Okay, can you see the web tool? Thank you. Okay, so when you go to the web tool, there's lots of really useful information. Actually, I'm going to copy at the risk of actually losing. Actually, no, I'm not going to do it. I was going to copy the address into the chat, but I don't want to mess anything up. So when you go to the web tool, lots of useful information, guidance and resources, frequently asked questions, and also case studies. Here's one that Sonya will be talking about in a moment. So go back there in your own time and have a look at some of the resources, but we're going to jump straight into the web tool. There's three steps. You identify your restoration site, select your species, and view your seed sourcing guidance. So what I'm going to do is, to identify your site, you can drop a pin on the map, enter the coordinates, or you can use this little search button at the bottom, which isn't working great at the moment. It actually takes you close to the site that you're, the area you've typed in, but you've got to know what you're looking for. Here's a travelling stock reserve near Berrydale. So you identify the restoration site, you view your climate layers, and this is all that NARCLIM 2.0 climate projections coming up. And the areas in blue are experiencing climatic conditions that are the same as what the restoration site are currently experiencing at the moment, whereas the areas in orange are future climate match. So they're currently experiencing what the restoration site is projected to experience in the future. So once you have your climate projections, then the next step is to select your species. Now, something I want to highlight, which is a common misunderstanding when people use the web tool, the intentionally long list of species that the web tool brings up are all the species whose extent of occurrence overlaps with the restoration site. So it's a very broad list, and you choose from that list the species that you want to restore based on your objectives and your knowledge of the site. So you can use resources like Trees Near Me, etc. So you get a long list of species. Let's look at Fenner for this one. You select it, it brings up all that genetic data in the background. When it's ready, you can click on View Collection Area. The first time you do this, it'll bring up this how to use information, which kind of just summarizes some of what I've just spoken about. And so then you view Collection Area. And what you can see for kangaroo grass, a very widely spread species, if you want to source material that aligns with natural gene flow in this species to safely maximize genetic diversity, you can source your material all the way from Moree south and then out west to Albury. So there's your genetic collection area for that particular restoration site for kangaroo grass. What I want to do now is I'm going to quickly jump to a different site. Okay, so I'm going to go to a site east of Jindabyne, which I have the coordinates for. 1, 4, 8, 6, 4, 6. Okay, it's a good idea to always check that it actually is where you thought it was. View climate layers. And then the next step, you want to select species. Now, one species I want to look at here is Bursera spinosa, or native blackthorn. Now, not all species behave neatly in a way that enables you to model their gene flow across the landscape. So for these, we have this additional information, map data not available. Doesn't mean it's not useful information, it's just different to the genetic collection area. So what we found with Bacteria spinosa is that the genetic data identified 3 distinct lineages. This is in New South Wales, and they don't align with the 2 morphologically defined species in New South Wales. And in some species, some areas, such as in the hunter, these lineages overlap. Whereas in other areas, which is more relevant down here in the south to the target restoration site, they don't overlap and they're consistent. So for this particular site, you would source your material only south of the Jervis Bay if you wanted to make sure you are maintaining those evolutionary lineages. Now, some species also have this additional button called Related Intensives. And so if you click on that, it takes you to a page where there's a report that someone's funded, it's been prepared. But it can give you useful information. This is for the Cumberland Plain Knowledge Infrastructure, which included Bacteria spinosa and, okay. Again, here's a little, the figure of the distinct genetic lineages, here the green down south. But what these reports have done is they've actually crunched the numbers and to give you an idea of the collection effort required to capture maximum genetic diversity, in this case, 90%. So for this particular species, Bacteria spinosa, crunching the numbers, if you want to capture 90% of that genetic diversity across a lineage or a genetic collection area, you want to source material from 8 sites, and in each of those sites, 5 mother plants, so 40 maternal lines in total. Actually, one thing I just want to highlight is if you don't maintain your maternal lines and you pull your seed from each site, the numbers tell you that you need to actually go to 22 sites to capture that genetic diversity. So by keeping, because you don't know whether that bag of seed that you collected from 10 plants, whether or not the germinates or the seedlings you have in the nursery are from one mother or from 20 mothers or 22 mothers or whatever. So for this particular species, for a genetically optimized seed sourcing strategy, they're the numbers. And so that just gives you an idea. And I hope, Magdalene, have I got like 3 more minutes?
You're all good.
Okay.
You've got 10 minutes.
Oh, wow, this is good. So what I want to show you is I want to go through a particular example rather than focusing on just quickly looking. We're going to we're going to go back to our Elizabeth Brownlee Reserve was a name I couldn't remember without my notes, sorry. So we're going to go back to that restoration site. And I just want to give you an example. So I'm going to use the Noble Road. Albion Park. Okay. Okay, and I've done a quick desktop. Many of you will know the target restoration site and may have a better idea of what your objectives would be. Where if I was to look at restoring subtropical rainforests to that site, I just wanted to give you an idea of how many species the web tool has in it for this site, for this vegetation community type, because we have done a lot of work on rainforests. So I'm just going to quickly select from this list. So again, as I mentioned, it's not all these species are relevant at all for that particular site. You select the species based on your understanding and your objectives. Okay, this won't take long. Okay, nearly there. Okay, and so once you've selected all the species that are relevant to you, thought I only got 10 last time. I probably accidentally clicked on something that was really unsuitable. you can actually go through and look at them one by one, but you can also generate a report, which sends a report either to your email or download, which then it's a really ugly report, but it has the information in there. Okay, save. So what it does is it brings together all the species that you just selected that are relevant for that particular restoration site in the Illawarra. There's a target site. And for each species, it gives you a little bit of information on, what we found and why. And then it maps the genetic collection area for each of those species. So if you scroll, and it also gives you, for those that couldn't be mapped, it gives you some additional information. So for example, I want to get to Microlina. Here's one, Dodonea viscosa. A little bit more messy, but it gives you an indication of where those different genetic lineages were found. They're not necessarily something, there's a lot of complicated by polyploidy and overlapping lineages and lineages that haven't yet been recognized. So it's very common for the genetic results to not align with subspecies. So we always look at an entire species when we're doing this work. We don't do it within subspecies. And I just wanted to bring up Microlina. And there's other species, for example. Oh no, I mustn't have clicked Microlina. So at the end of the report, it actually gives you all the combined genetic collection areas. And that is a very super quick overview of the Restore and Renew web tool.
Thank you, everybody, for sharing your Thursday evening with us. It's great to have so many people online. And I'm joining you from Ngunnawal country in Canberra tonight. So I'm a project officer with the Yass Area Network of Landcare Groups, or YAN. I'll call it YAN from now on. And I work specifically on the Climate Ready Revegetation program. We're lucky to have a 4-year New South Wales Environmental Trust grant that's funding my position for 2 days a week until next spring. I'm also involved with the Southeast Climate Ready project, so working with Maggie and others to organise this webinar and the follow-up sessions. So I want to say up front, and it's probably the main message from me tonight, is that climate ready revegetation won't look the same everywhere. And I think Tricia's given us a taste of that with those different environments and different options. So the underlying principles that Tricia has introduced remain the same, but how you put them into practice will depend on your particular situation. So what I'm sharing tonight is how Yan has approached it, and it's just one practical example. I'd like to acknowledge that in the Southeast region, and I'm aware Upper Shoalhaven, Upper Murrumbidgee, Bungendore Landcare, they've also applied climate ready concepts. And certainly there are lots of groups in our region with extensive experience and expertise in collecting seed and propagating plants, both in nurseries and through grow at home models, experience in revegetation, seed banking, and even setting up a living seed bank in the case of the far south coast. So I think we're in a really good position to learn from one another about all these different aspects and how we can integrate climate ready concepts and principles. So the YAN program reflects our people, our regional ecology, and our existing infrastructure and practices, and the information that we've been exposed to at different points along the way. So to briefly introduce our people and place. When I speak about this program, I'm speaking on behalf of the YAN Climate Ready Revegetation team. It's an amazing group of volunteers who started this program back in 2018. And they've put hundreds, if not thousands of hours of work into it, and they're all still involved 8 years later. And more recently, Sarah, the coordinator, and myself are also part of that team. To locate where we are in the landscape, obviously Yass is just north of Canberra, of ACT. The Yan area roughly aligns with the Yass Valley Council area, which is outlined in white there. The Landcare groups in our area include Bowning-Bookham, Yass, Murrumbateman and Sutton, and we also have the Friends of Yass Gorge in our network. We currently have 3 volunteer-run Landcare nurseries in our area, and the Murrumbateman group also has a relationship with the Alexander McConaughey Centre, down in Canberra. So that's an adult correctional facility in Canberra, and they grow plants for us that we distribute in our area, specifically through Murray Bateman. And this is a photo of, well, some photos of our nurseries and nursery volunteers. Over the course of a normal year, there are something like 75 volunteers who help out at our nurseries, and well over 2,000 volunteer hours that go into all aspects of plant production and nursery maintenance. So why climate ready revegetation? One of the key factors that brought this group together and kept the program going for 8 years so far is a common concern about climate change and its impact on our local environment. As we've heard tonight, climate change threatens the survival of native plant species and the longevity of traditional revegetation efforts. Like in this photo, kindly supplied by Maggie's group at Upper Snowy, the dieback event of Eucalyptus viminalis was a big wake-up call for people in our region, because it demonstrated the vulnerability of even an iconic keystone species, and it's quite shocking to see these sort of images. I think I'm probably speaking for a lot of people that climate change is big and daunting, so it's empowering to be working on something that's positive and practical as a response. Just to reflect briefly some of the other key factors that have got this program off the ground and has kept it going. I've spoken about that strong motivation and commitment. In 2018, our group was lucky to have a workshop with Nola Hancock and Linda Broadhouse, and I think we're really lucky to have Nola online tonight. Thank you so much, Nola, for joining. They just published the Climate Ready Revegetation Guide, and that was a big boost at the start of this work. We've also benefited from other external expertise. In the early days, Adapt New South Wales was vital. They provided downscaled climate projections via ANU. Luckily, that information is now publicly available, which I'll touch on later. We have a very strong team approach to our work with complementary skills across our team. And importantly, in our area, we had existing Landcare nurseries. So we were already producing plants, and it was a matter of adjusting some established processes rather than starting from scratch. And as you can see from that photo, we do try and make our meetings enjoyable. We share a meal as well as having some pretty robust conversations. The aim of our program is to enhance the long-term survival of native plants in the Yass Valley region of New South Wales under changing climatic conditions. So far, we've been mostly focused on tube stock production for local revegetation efforts to make sure that the revegetation plantings in our area will have the greatest chance of surviving and forming self-sustaining populations in the long term, just like Tricia was explaining. So our process can be summarised in these 3 steps, which I'll go into more detail after I give the main headings here. So firstly, species assessment. We choose which species to focus on, and for us, we've chosen to choose species that are most likely to survive because they occur in hotter places, and I'll talk more about that later. And then for those species, we maximise genetic diversity, so they have the best chance of adapting to changing climatic conditions. And then we do that by sourcing seed from a range of provenances. And then our nurseries propagate the plants, landholders plant and monitor. So I'll go through these steps in a bit more detail. When we assess a species, we ask, does it occur locally and in areas that experience our future projected climate? And we determine that through mapping and analysis using publicly available data and tools, which I'll introduce a bit later. We ask, is it ecologically important? Is seed commercially available? That's because by and large, we purchase seed from retailers, and I realise that that's not the case for a lot of other groups. We also ask, can it be easily propagated at our Landcare nurseries? As a lot of you will know, some species are much easier to propagate than others, so we have to be quite realistic about where our volunteers, how they spend their time, what they grow. So once we've got the seed, once we've chosen the species, we then source the seed from a range of provenances to maximise genetic diversity. Now here, our original approach was to source seed from across a species distribution. And here, with the example of yellow box, Eucalyptus meliodora, the black dots show the distribution of the species, and the red dots show where we've managed to find source seed from. So it's pretty, I think, good representation from across that distribution. Now, of course, more recently, we've been exposed to Restore and Renew, and we're now informed by that genomic research through the Restore and Renew web tool. So this is for the same species. It shows that recommended collection area. So it's tightened it a little bit for this species. But we find this incredibly helpful. It covers about a quarter of the species on our nursery list and most of the eucalypts. It gives us confidence in our seed sourcing decisions, and we have certainly adjusted our practices in light of this information. It doesn't cover all the species that we're interested in, but we'll continue to adapt as that information becomes available. So that's sourcing the seed, and then we grow the plants. So we grow about 40 species in the nurseries at any one time. That's because the nursery managers have realised it's a good diversity of species while also being manageable for the operation of the nurseries. In total, about 15,000 tube stock are grown every year and distributed to landholders for planting. And most years we have about 100 landholders who get plants and plant, which is another huge effort. And we estimate that something like 1,500 hours of time goes into planting. That doesn't include fencing and ongoing maintenance either. So another huge contribution from landholders. We do encourage our landholders to monitor so that we can continue to improve our practices. And in a little bit more detail, we have a 3-species trial investigating whether there's a difference in the 3 to 5 year survival rate between plants grown from seed source very locally compared to seed source much further away. And that trial was established in 2021. And so far, the results show that there's no real difference in that early survival of those 2 treatments. We also have a wonderful partnership with the RESA team at Botanic Gardens of Sydney on genetics, and that's supported by the current grant from the New South Wales Environmental Trust. So in this photo, this is Tricia's colleague, Marlene Vandermeer, who was in some of Tricia's photos as well. So here, Marlene and the volunteers are collecting some leaf samples of Eucalyptus meliodora. These samples were genetically sequenced, and we pose a question of whether our seed sourcing strategy is achieving the genetic diversity that we're aiming for, and what else could we be doing? So we asked that of the scientists, and just in the last couple of weeks, we got the report back with their findings. And just from sort of really just skimming it so far, the results show that we are indeed enhancing genetic diversity by using our seed sourcing approach, but we're also seeing some of those dilemmas that Tricia's mentioned in regards to hybridization, misidentified species, and high levels of relatedness. So we'll be meeting as a team to digest all this great information and trying to work out what to do from here in response to that. I wanted to also just quickly introduce a couple of the wonderful publicly available tools other publicly available tools that we draw on in our work. My Climate View has become available since we started the project. So where we needed to get sort of tailored localized climate projections, now anybody can access this. It's a collaboration between CSIRO and the Bureau of Meteorology. It provides historical climate data and downscaled projections at a 5-kilometre grid resolution. So it gets pretty local, and it's very helpful. So what we do is we get the projections through this website now, and what we can do with that is turn it into maps. And we also use this other great resource, Atlas of Living Australia, which Matt mentioned in his presentation. So what we can do is map our future climate area. So in this map, the red dot is Yass, the orange-brown shows the areas in Australia that currently experience the range of temperatures projected for Yass by 2070 under worst-case emissions scenario. And obviously you can change those variables. The blue shows the area that currently experiences the range of annual rainfall projected for Yass. And it's where those two overlap is the future climate area. And this is really similar to what you can generate in the Restore and Renew tool that Tricia demonstrated. One advantage of doing this through the Atlas of Living Australia is that you can also then layer it with species distributions that aren't yet available on Restore and Renew. So going back to our species assessment, we use these maps to check whether a species occurs in our future climate area. And for those species we want to investigate a bit further, we also generate scatter plots. So this is the same information presented just a bit differently. So here the black dots are the occurrences of Eucalyptus meliodora. On the x-axis we have annual mean temperature, on the y-axis we have annual precipitation. The red dot is current Yass climate, the blue dot and rectangle are the future projected climate. So we can see from looking at this that this species currently occurs in places with climates similar to those predicted to Yass in the future. So we take that as evidence that this species has a good chance of tolerating our future conditions. But I do want to highlight that just because a species doesn't currently grow in hotter places, it doesn't mean that it won't survive hotter conditions. And Tricia might like to speak about this more later. And some groups might even purposely choose species that are assessed as more marginal so that they can boost their genetic diversity to help them to survive and adapt. So that's another perfectly valid approach. So future directions for our program. The team's keen to increase our impact. One way we're doing this is through trialling some direct seeding of our multi-provenance seed. The Bowning Book and Landcare group is doing this through their Habitat Hops project for the first time this year. And also through spreading the word and introducing these concepts, introducing Tricia to more people throughout the Southeast. So that's what we're doing with this project. We're also interested in securing genetically informed seed supplies. If we get more funding, get the chance to do this. Rather than being at the whim of the market, we can imagine perhaps strengthening relationships with seed collectors, make sure they're collecting the right material from the right places, and also investigating the possibility of setting up one or more seed production areas, which we could make very sort of carefully curate what plants go into them to be confident about the seed that's coming off them. So they're just a couple of things that we might be doing in the future. So what can your group do? Hopefully the takeaway from my presentation isn't that every group needs to copy what Jan has done. The takeaway is that there are practical ways to bring these ideas into local revegetation work. And you might just be starting with conversations with your local groups, your local seed collectors. You might just start with one species, one planting site, or one change in your nursery practices. The important thing is to start applying the principles in a way that makes sense for your group and your landscape. And if you're keen to see some more examples of how to apply genetically informed climate ready principles, I'll paste a couple of links in the chat after I finish to the Big Scrub Genetic Rescue Project. It involves Tricia's team at RESA, and it's really inspiring. It's based on some rainforest up in North New South Wales. And that's pretty much all I wanted to say. There's more information about our work on our website, and we can go through a lot more detail with those groups who would like to take the next step in this project in the Southeast. So thank you, and I'll shut this down now.